Today we discuss about a very important hormone, that is vasopressin. Vasopressin is a small peptide hormone. This is synthesized by supra-optic and paraventricular nuclei in the hypothalamus. Oxytocin is also secreted by supra-optic and paraventricular nuclei in the hypothalamus and later travels to the posterior pituitary. So after synthesis, the hormone stored in posterior pituitary and ultimately enter the blood. Now, when vasopressin is secreted from the hypothalamus, H2O deficit or concentrated ECF means when your body faces H2O deprived condition, your extra ECF means extracellular fluid becomes hypertonic, concentrated. So you have to preserve or conserve your H2O. You can't lose much H2O during urination process. So in this video, we discuss about the role of the vasopressin in kidney. Vasopressins have different functions, like it control our sexual behavior, attention, learning, memory, partner selections. Our anxiety level decreases in presence of vasopressins. Blood pressure increases in presence of vasopressin. Blood vessels contain different types of receptor, that is a V1 receptor, and when vasopressin binds with the V1 receptor, it helps in constrictions of that blood vessels and ultimately blood pressure increases. So there are several functions of the vasopressins, but today we discuss only the role of the vasopressins in kidney tubular cell in water reabsorption in H2O deficit condition. So we take a cell, that is tubular cell. These cells may be from distal tubule or collecting tubule. So first point says basolateral membrane of DCT and CT contain V2 receptor. In case of kidney, the vasopressin receptor is known as V2 receptor. So this is V2 receptor. And so basolateral, so first we have understand what is basolateral and luminal surface. This side of the tubular cell means the side which faces towards the peritubular capillary is known as basolateral side. On the other hand, the side which faces towards the filtrate known as luminal surface or luminal membrane. So basolateral membrane of DCT and CT contain V2 receptor. As you can see, this is basolateral membrane. This basolateral membrane contains V2 receptor. It is a G protein coupled receptor. G protein coupled receptor. Okay, so there is a G protein that helps in the activations of several other proteins. So vasopressin binds to this receptor. Okay, vasopressin binds with the receptor. Hypothalamus secrete hypersynthesize the vasopressins. Then it is stored in the pituitary and ultimately enter into the blood and comes to this V2 receptor. But when your body in H2O deficit condition. Now next point says cAMP is formed from ATP with the activation of adenylate fusion protein that helps in the water channel fusions in the luminal membrane of the tubular cell. These are aquaporin 2 receptor vesicles. So when cAMP is formed inside the tubular cell, it helps in the fusions of water channel in the luminal membrane. This water channel aquaporin 2. Now cyclic AMP helps in the fusions of aquaporin 2 channel in the opposite luminal membrane, increases its permeability to H2O from filtrate. Our body needs H2O. We can't lose much H2O during the urination process. So we have to collect that H2O from the filtrate. The H2O where the H2O moves towards the tubular inside the tubular cell through this aquaporin 2 receptor down its concentration gradient. Now from the tubular cell it enters the interstitial fluid. As you can see, this is interstitial fluid, and this is peritubular capillary, and ultimately H2O reaches to the reabsorbed in the peritubular capillaries. The concentrations of H2O in the filtrate is more than the concentration of H2O in the interstitial fluid. The interstitial fluid, in case of distal tubule, the interstitial fluid concentrations near about 300 milliosmoles per liter. On the other hand, the filtrate concentration is or you can say osmolarity 100 milliosmol per liter. So water moves down its concentration gradient from filtrate to the interstitial fluid and ultimately reabsorbed into the peritubular capillaries. Now next point says that H2O enters the tubular cell and passively leaves the cell down its concentration gradient across the basolateral membrane to enter the interstitial fluid. Ultimately, water is reabsorbed into the peritubular capillary. Water channels in the basolateral membrane are vasopressin independent. So the water channels which are present in the basolateral membrane, these water channels are always present and they are not dependent on the vasopressins. On the other hand, if we remove the vasopressins from this V2 receptor, the fusions of the aquaporin 2 channels does not occur. So these aquaporin 2 receptor which are present in the luminal membrane in the tubular cell depends on vasopressins in H2O deficit condition. When we have excess H2O, the vasopressin does not bind and there is no channels in the luminal surface. So aquaporin 2 depends on vasopressin. So this is the functions of the vasopressins, and in the later video we discuss about the detailed how vasopressin works in oxygen H2O deficit conditions, or is vasopressin does vasopressin any effects on the distal tubular collecting duct during the when we have excess H2O. So in the later video we discuss it. So the main functions of the vasopressin is to reabsorb the water from the filtrate and ultimately this water enter into the peritubular capillary. So main functions of the vasopressin is reabsorptions of water from filtrate. If you like the video, please share with your friends. Thank you.